So hello, welcome to our final lecture series in clinical biomarker research, and we're going to be talking about gene expression assays for biomarker discovery. I kind of went a bit not in the traditional order of the central dogma, which you would think I would end with proteomics and metabolomics, but hopefully this still makes sense in the context of how we are looking for new biomarkers and how we try and assay them. And this is by no means, as I always say, a comprehensive lecture. We could probably have an entire year series on just gene expression and each platform that you can use and how to analyze it. So it's just giving you a little taster of what you should think about, what we can use and how they can be used in the clinical context. So measuring gene expression can be quite powerful in terms of identifying how cells respond in different states. And we know that the formation of functional protein starts with the transcription of coding genes into a mature mRNA molecule. And we know if we look at the genome, all cells will contain the same amount of genes or the same set of genes, but not these genes are not necessarily expressed in every single cell type or under different conditions. And so gene expression in an organism is a very dynamic process, and it can change for a multitude of reasons and in different tissue types or different cell types. So for example, the same gene is expressed differently in different conditions and different cells, and we know that there are other processes that happen like gene editing, modification, alternative splicing that result in different final end products, which is our proteins. So if we look at just our normal central dogma, we know that there are exons and alternative splicing that happen, so even though we have the same genes, at the end of it, depending on the alternative splicing event that has happened, you will have a different functional protein. And this is all regulated in our cells under different conditions, and that's why it's incredibly important to be able to measure gene expression in different cell types. So being able to quantify levels at which genes are expressed in cells, tissue, or organisms is also known as transcriptomics. Right? So this is in traditional genomics, we're looking really at gene expression. And here you're measuring the transcriptome. And traditionally it was thought that really looking at the coding portion is the most important part, right? mRNA makes proteins, and so a lot of the focus was on the mRNAs. But now we know that actually the non-coding part are equally as important. So things like your ribosomal RNA, your tRNA, your short and long non-coding RNAs have regulatory functions, and they're increasingly becoming more important in terms of looking for different biomarkers for gene expression. So if we look at the available methods that we have, it's very similar to our proteomic strategies. You have two main categories. You can look at targeted gene expression. So here, very old school technique now, which is northern blots. So you're looking very much at single genes at a time. I don't know if anyone's ever done a northern blot. It's very similar to a western blot, but it's quite a long process to look at just the gene expression of a single gene. RT-qPCR, you can say it's a bit more, you can look at multiple targets, but it's still considered a targeted approach. And now we're looking at, and we're entering, as I always say, the omics generation, where we're looking at the global untargeted transcriptome. So if we look at global analysis of gene expression profiles, there are two main strategies that people have looked at. And the first is gene microarrays, and I'll go in a little bit into the strategy of how gene microarrays work. But here you basically have probes that are immobilized onto a surface. And the problem with gene microarrays is that you need a priori knowledge or previous knowledge about which genes you're going to target. So you could say that this is in fact not truly untargeted. You're not really looking at the global gene expression because you need to know what you're going to look for. And it does have limited dynamic range, and we'll go into why that is the case. And we've now entered really exciting platforms in terms of being molecular biologists with Rna Sequencing. So it's a whole transcriptome shotgun sequencing that are able to look at the entire transcriptome as it is in different cells, in different tissues, and even in single cells. And we'll go into those platforms and how they can be utilized. So gene microarrays, they're pretty old technology, but they are still routinely used in many diagnostic labs. So you can look at gene expression of sequences that you already know. So you can, on a single plate, look at about 1,000 genes, so it's quite high throughput. You are using DNA fragments, which are your probes. They represent the coding region of your genes, which are immobilized onto the surface. So if we imagine here, we have the control in the experimental group, and I'm sorry, I should have a pointer. When you convert your RNA into cDNA, they are subsequently labeled with dyes. So your control in this case is green, your experimental is red. and then you do a hybridization onto your plate, which has these probes that can recognize these targets. After doing your hybridization, you can then express your fluorophore, so either green or red, and depending on what comes up on the plate, so in this case you can see, if you look at the intensity, so on that first one you would have just a red, which shows you that you have this upregulation of the gene. If you've got just green coming up on your one gene sequence or on your probe, then you know that there is a downregulation, and if it's black, so both of them are cancelling each other out, then you've just got constitutive expression, so no differential regulation for that gene. So this has been quite a powerful tool, and you can do thousands of genes at the same time for 2 different samples. But as you can see, it's definitely quite a laborious process. The fact that you need to know the genes that you're looking at really makes it that you're never going to look into discovery-based gene expression, so you're not going to find new targets. And there are problems with things like cross-hybridization that happen on your plate that have made things quite complicated. It's also very difficult to look at lowly expressed genes, so usually a microplate has very limited dynamic range for gene expression. So this takes us into kind of the next generation of how we're looking at gene expression, which is RNA sequencing. And for gene expression, we're looking at whole transcriptome shotgun sequencing, so WTSS, which looks at the entirety of the transcriptome. So we're looking at the mRNA, which is the coding portion, but also non-coding splicing events and PTMs. So it can be quite powerful to look at the entire transcriptome and what's happening in a cell that's under a certain condition. You can look at things like mutations, single nucleotide polymorphisms, insertions, deletions, copy number variation, which is also very interesting, chromosome rearrangements. So you're getting a huge amount of information with this new technology. And it's really, I think, the way biomarker discovery is moving for gene expression is in whole genome sequencing. Costs are coming down. It's quite easy now to perform in most labs. And I think this is really becoming the future for gene expression. Another advantageous platform for RNA sequencing is that you can start with a very small amount of material, so you only need about 100 nanograms. Microarrays need much more RNA to do this. And from just a tiny amount of RNA, you can get a huge amount of data. And there's a very large dynamic range. It's a very sensitive platform. So I think in terms of discovery-based, if you can do an RNA sequencing project, that's really the way you should be going. So I wanted to describe kind of a basic workflow of Rna Seek, and there are different strategies that you need to employ depending on your tissue type, your cell type, and of course the experiment itself. But basically your first part of the experiment is library preparation. So you're going to do an RNA isolation, very standard RNA isolation. It will change depending on what you're working on. But you're going to do something like DNase treatment. You need to assess the RNA quality, right? So usually we use RIN numbers. Quantify how much RNA you're able to get out. And then we look at RNA selection or depletion. So in your transcriptome, when you've extracted all of your RNA, 90% of your RNA is ribosomal RNA. So this is quite problematic, of course. So you can look at your transcriptome as is, with your ribosomal RNA, knowing that perhaps you could miss certain targets that are perhaps lowly abundant. You can also do a poly-A selection. So polyadenylation happens to mature mRNAs, so you can use poly-A selection to just pull out the coding part of the transcriptome. You can also do a ribosomal depletion, where you get rid of the rRNA and keep everything else. Now, can anyone see where issues would happen with each of these techniques? So say you're looking at ribosomal depletion, what could happen to your downstream RNA that you collect? Yeah, so you could have like non-specific depletion that happens. So you actually end up pulling out other RNAs that maybe stick to ribosomal RNA. So with any of these strategies, if you're going to do a selection or depletion, you need to be cognizant of the fact that you're introducing minor biases in any of your downstream analyses. But it is a good idea to read around the subject and decide before you start collecting your samples how you're going to treat your RNA for your Rna Seek experiment, because each of these has their pros and cons, and you need to weigh up what you're willing to sacrifice. After you've done your RNA isolation, say you've done poly-A selection, you're going to convert to complementary DNA, so it's reverse transcribed, and then you need to do sequence purification and just removing low quality sequences. So at each of these steps, you could be introducing errors and different steps, and it's very similar to any of these proteomics assays that we do. And it's always good to read around, understand where you're introducing problems into your workflow. Finally, then you're going to use any next-generation sequencing platform, and that is entirely up to you. There are a number of companies that now do this, that will now sequence your cDNA. And then comes possibly the most important and tricky and horrible part of your experiment, which is the analysis. And we'll go into a little bit of the workflows people use for RNA Seek analysis. So first is the transcriptome assembly. And you can either do this with sequence guided, so if you have a database that you can query against, you use that. If you're working on a non-model organism, and you're very unlucky, you're going to have to do a de novo assembly, right? So you can either do sequence guided or de novo, and each has their own pros and cons. Of course, with the sequence guided, you are almost targeted against what is available in your database. So sometimes de novo is better, but de novo can be more complicated and give you results that maybe aren't so clear-cut. After you've done your assembly, you need to look at differential gene expression. And typically people do counts. So if we look at our experiment here, when you've got first sample and the second one, I feel like I need to move to show you guys. Then you've done your RNA Seek, and then you essentially just count the number of reads that have mapped to the gene. So if we're looking at gene A, which is in red, in our first sample, there are 6 counts for gene A. And then in sample B, so let's say our experimental one, you have 5 reads that have mapped to that gene. So the ratio of differential expression from the first sample to the next is 1.2. So you do ratios of differential expression to show if you've got an upregulation or downregulation. And that's a very typical way of doing differential gene expression for RNA-seq data. After that, you really want to look at co-expression networks, functional analysis. I think I've shown it here, where you can do co-expression of different map reads. You can do network constructions and see which pathways are being activated or downregulated or being affected. Module definition with hierarchical cluster analysis, so which genes are changing together. And then there's things like guilt by association, predictions, and things like that that you can do. So you've got a huge amount of information, and now your analysis is going to be an enormous task to do. And it's good to have a good plan. And I've tried to include different publications on each slide that you can have a read through that really discuss this in detail. I'm really touching the surface here on what you need to do for analysis. So keep talking about bioinformatics specifically because this is a bioinformatics group. So bioinformatics for your RNA-seq or even any gene expression is going to take up the bulk of your time. It is the most complicated part. It is highly, highly complex. And I think one of the problems is that with any new technology, the tools have not been established and well validated, so people are constantly coming up with new tools, and you almost become so overwhelmed with what you should do. So there are 2 blogs or 2 resources that I've tried to put there, the Rna Seek blog and Rna Sequencing, which try and consolidate all of the different tools that are available. So I really encourage you to explore these blogs. I'm sure a lot of you have, Marina, you've probably seen it. They really detail each method that you can go through, what the pros and cons are, and exactly what you need to take into account. I've tried to show the different pathways that you can take. And one of the biggest things with the RNA-seq is quality control, like your QCs each step. So you need to QC your raw reads, you need to pre-process, filter, trim, then you need to QC the mapping, then you need to QC your counts, then you need to have the right stats in place. So I encourage anyone to talk to a statistician or someone who has experience with RNA-seq. And then you need to do things like multivariate statistical analysis, looking at your transcriptome-wide differences, and this is all highly complex. And like I said, your tools are constantly evolving. So you need to make sure that you're up to speed with what the latest tools are, what they can do, and how to interpret this. Because at the end of the day, we're all trying to get biological insight into what's happening. And if you incorrectly analyze your data, you're not going to get the correct answer or actually what you're trying to discover. So it's important to understand each of the steps and how they affect the downstream analysis, which I have something about assumptions that we'll talk about in just a moment. So let's talk a little bit about practicalities behind sampling design. A lot of the times, if you're part of a clinical trial, and I think for bioinformatics team it's quite difficult, you haven't had a say in how the sample is collected, but it's good to be aware of where things can go bad on your RNA. So when designing your sample, your experiment, what is your research question? What is it that you're trying to find? Because if you're going to look, say, just at the blood, but your disease only affects your spleen, perhaps you're just going to miss any differences. So you need to be aware of what are the tissues that you're going to target, can you access those, and is using a proxy like blood appropriate? Talking about homogeneous sampling, so taking samples from the same demographic, same age, things like that, or are you going to have heterogeneous sampling? Things like that can affect what you see at the end of it in your biological inferences. Replicates, biological and technical, I feel like there's still some people that have issues. So what is the difference between a biological replicate and a technical replicate? I'm throwing this back out.
Oh. So a technical replicate is, or let me start with biological, is from the same condition having, say, 3 different mouse
Yes.
or mice. And technical would then be taking a bunch of different samples from the same mouse and analyzing them in exactly the same way to see how much variation there occurred.
Yeah. Thanks. So if you're taking a sample, your blood sample from the same mouse, and you aliquot it into 3, and you take the same measurements from this aliquot, this aliquot, this aliquot, is that n equals 3? From that same mouse, you've taken 3 samples. The same one. It's 1. And I think sometimes this happens, people think technicals can count as biologicals, and it's important to know when to use technicals, when to use biological. Ideally, you can do both. So in your multiple biological replicates, you also have multiple technical replicates to account for variation, inherent variations of experiments, you working on that day. Oftentimes you can't do this, so it is better to have biological replicates for an experiment. It is better. But if you can, try and include technical replicates, especially for the downstream validation parts. It becomes important. So when choosing your research questions, you also need to think about RNA yield. Some samples just do not give you enough RNA. And Rna Seek doesn't need a lot of RNA, but if you're only going to be able to scrap out a few nanograms, you're dead in the water from the get-go. So understand what tissues, and I think Tandeka knows the heartache of working on a sample that will not yield you enough sample. It's devastating, so definitely before you plan a huge project proposal on roots of the banana leaf, find out can the roots actually give you RNA. Right, so RNA is a very labile molecule. It is short-lived, it wants to degrade, there are RNases all over the place. So when collecting your sample, you need to think about how best you're going to stabilize your RNA. You can do something like snap freezing immediately. So after you've harvested an organ from your mouse, it needs to be put in liquid nitrogen and stored at -80. There are also a lot of stabilizing buffers, RNA later, triazole, that kind of thing. I think I wrote RNA later twice, apologies. There are a number of RNA stabilization buffers that you can use just to stabilize your RNA and make sure that at the end of your 2-year clinical trial, your samples you collected 2 years ago aren't degraded. So it's very important to be able to think about how the RNA was preserved and is the RNA still good quality, especially if you're doing the bioinformatics analysis downstream and you're like, what's going on? These samples, go back and see how they were collected. So the success of your Rna Seek experiment is dependent on your pure and intact RNA. At the end of the day, we always say crap in, crap out. So make sure that you have good quality RNA from the get-go. Act fast. Be cognizant of the fact that there are RNases everywhere. You need RNase-free tube, RNA zapper, inactivate your RNAs, work in a clean hood. Just make sure you're stabilizing your RNA. RNA can be very problematic to work with. So I want to talk about the assumptions that we make when we're looking at gene expression or RNA Seek. So we're going to start from the bottom of this flowchart and move up. And this is where I think trying to infer biological insights about what is happening can become problematic. So we've now done our RNA Seek, and we've got RNA Seek reads, and the assumption that we're making is that the RNA Seek reads represent the cDNA pool that we've created. Now what is the problem with that assumption? The fact is that when you do your RNA Seek, there are a bunch of things that can happen, like failure to map the reads to the correct genes, biases of reading cDNA fragments that will affect the fact that that cDNA pool is not exactly a perfect representation of what we're seeing. From our cDNA pool, we're assuming that the cDNA pool represents the RNA that we've extracted. Another problem here, you can have loss during your extraction. You can have loss because you've done depletion or selection. Your conversion of your cDNA was not 100%, so some things were missed. So another step where we're making incorrect assumptions that could have an effect. From our own RNA, we are assuming or making the assumptions that mRNA levels are a proxy for protein activity. But we know that we have mRNA templates that have different speeds of regulation or protein production. mRNA is degraded at different rates. You have alternative splicing. So that assumption is also not 100% true. We also assume that the proteins that we identify from this assumption, this long list of assumptions, defines the phenotype. But we know that proteins undergo PTMs, phosphorylation, ubiquitination, and differential regulation. So in fact, can you see that all along the way with these assumptions that we make, that is perhaps why at the end a lot of these targets fail. And because we make assumptions all along the way in our experiments that not necessarily are 100% true. And I think it's hard to get around that. You know, we always ask ourselves, like, how do we make sure we have the best experiment and that we are going to find these targets? And I think being aware of where you're making assumptions and where things are not exactly what you think they are, and when you talk around it and when you try and infer hypotheses, just have that in the back of your mind, that things are perhaps not as clear-cut and you cannot make these assumptions. And I think that's the best way to protect yourself and really understand and not overestimate what you're describing, what you think you found. So I want to briefly talk about kind of the next level of Rna Seek, which is now single-cell Rna Seek. And some people already in this department are looking at single-cell Rna Seek analysis. If we look at bulk sequencing with Rna Seek, we are taking the average expression profile for all the cells in our sample. And the problem is that cells are there's a lot of heterogeneity. And if we are looking at this bulk population of cells, there could be individual cells that are actually having a real effect and a real change. But because we're looking at the average, we're going to fail to identify that change. So this is where RNA profiling of single cells can be very exciting and can give you quite a lot of data out of it. So here you're profiling single cells, usually about hundreds of cells. I think you can go up to 1,000 if you have a lot of money. And you're going to isolate your single cells, so you can do it from your tissue or your cell suspension. So there's things like fluidigm, microfluidics, sorting with flow. You can do laser microdissection, where you cut out individual cells and put them in your tube and go and analyze them with Rna Seek. And what's very exciting with single-cell Rna Seek is you get the spatial and cellular context of these cells in their transcriptome in this environment. So I think it's still quite tricky to do a single-cell Rna Seek experiment, but you can get quite a lot of information out of it. And quite a lot of people are starting to do it, and it's just something, I think the analysis for it also has its own pros and cons. So think about it if you are designing a new experiment, about the fact that bulk sequencing also has biases in it, and you may want to look at single cells if you're looking at a specific pathway. So finally, I want to talk about validation. So why do we need to validate? Any experiment, proteomics, I spoke about validation too. What is validation and why do we need it? You've now done your experiment, you've done your Rna Seek, and you're like, oh, gene X is differentially expressed in my condition, it is a biomarker, I'm ready to go for clinical trials. Why do you need to validate your discovery-based experiments? Right, so how should you validate an experiment? Can you redo Rna Seek on your experiment? Is that validation?
You want to use a different technique or a different instrument to measure something about samples and see whether that finding is actually true.
Exactly.
Whether it holds in the second time around.
Yeah. So does this now hypothesis gene X is a biomarker, does it still hold true in an independent technique? Because sometimes there are aspects of a certain technique that you've done that are going to give you this result, but it is not true in another independent technique. So if you look at RT-qPCR or you do in situ hybridization, you can do functional validation, you need to verify, validate that these measurements are in fact true and that they are increased in your disease condition with a completely independent technique. You cannot use an Rna Seek on the same data and say I've validated it. That's more verification. And so there are a number of ways to do this for Rna Seek. Functional validation is probably your best bet at actually really identifying how the pathways change. You can do gene knockdown or rescue experiments, and I think it's nice if you can do a temporal look at your gene of interest. So if you think about it, we take snapshots of cells, but in fact maybe a gene expression actually really comes up much earlier than what we think. So it's always a good idea if you're going to do an RT-qPCR or a proteomics experiment, broaden your timeline, because now you can, because you're doing a targeted assay, and then you can look much more comprehensively how this gene operates in your cell and your condition. Of course, you're limited by the sample that you can get, so that is something you have to keep in mind. So validation is very important. It's always important. I would say you always have to validate your experiments. Some papers seem to get away with not doing it, but you do need to do it, especially if you don't have that many biological replicates. Then you really, really need to have solid validation for you to make a true statement about what you found. And also your target choice is important. So if you've got poor sequence depth on your Rna Seek, maybe that's not the best target to look at. Is the pathway relevant for your disease? Maybe this gene came up, but it's got nothing to do with the pathway, and it actually maybe maps to something else. And you need to, ideally, if you can, conduct a whole new set of biological replicates to ensure that's true. A lot of people validate on the original data set, but I would say to have a very robust validation that you conduct a new set of experiments or a new cohort to validate what you found. So in terms of gene expression assays are being used in the clinic, it's really becoming, well, it has been very commonplace for a long time. So I just wanted to use a few experiments. So for example, pathogen detection, using qRT-qPCR, looking at viral RNA, influenza virus, dengue virus, HIV is probably the most common one. You can now use at day 9, look at the HIV viral load, looking at the viral RNA of HIV and identify if someone is infected. Ebola is centered around RNA of the Ebola virus. There's things like looking at the mRNA signatures, AlloMap. It's a non-invasive gene expression blood-based signature. So when a lot of patients, when they need a heart transplant, some patients, for some reason, reject the heart transplant. And they've come up with this new gene signature that identifies patients and stratifies patients on whether or not they are at risk for rejection. So it has this quantification score around the blood-based signature, and it's being used quite routinely now. It's called AlloMap. And what gene expression and these Rna Seek signatures are giving you is this personalized medicine, because we're all looking to have these personalized biosignatures that will identify patients that has this or this condition or higher risk. And we're looking into this multi-gene fingerprints that are becoming really a reality now that we have the platforms that are coming down in cost and are highly sensitive to be able to provide personalized medicine and diagnostics. So this is not a comprehensive list of all of the tests that do this, but it is routinely being used in a number of pathology labs and clinics. So just to summarize what we've been talking about, transcriptomics offers insights on how gene expression dictates and controls cell physiology, how cells respond. Rna Seek is a versatile, comprehensive solution. It has high reproducibility and sensitivity, and you don't need any prior knowledge, so it's a very discovery-based, untargeted approach. The bioinformatics is your most challenging problem, I think, in your Rna Seek analysis. It's highly complex, it's ever-changing with the tools, so keep up to date with what's being released all the time. Your experimental design is equally as important, because if you don't design it properly, you're unlikely to see anything of value, or you'll waste a lot of time. And you need to consider the appropriate amount of biological replicates that you need to have enough statistical power to make a decision about whether or not your diagnostic gene expression or your genes are of value. Transcriptomics are being very routinely used in disease management, and it's really becoming quite a hallmark for personalized medicine. Thanks very much. Are there any questions?